from biomedical engineering, and on behalf of Chuck Tabor and myself, we welcome you all to the Provost Graduate Student Seminar Series. This is a series we established 2 years ago. This is our second year, and it is allowing the graduate students to present to a larger audience to get an idea of what it's like to present to the public at large. And our selection process is highly competitive. It will be my honor to see the lecture today. I want to make sure you guys know to eat all the food. It's already paid for, so help yourself. And now I want to introduce the person who will introduce Javier. It's Professor Akchakaya from Ecology and Evolution.
Yeah.
Thank you everyone for coming. It's my great pleasure to introduce Javier Monzon. I have been on his thesis committee for some years now, and he'll talk about the hybridization of coyotes and wolves, which is a very interesting topic. Now, hybridization is interesting in itself. Javier is looking at it from the point of view of the effect of ecology and geography on this process. And there are a lot of interesting science questions that he'll talk about. But I also want to mention that the topic is also important from a practical point of view, from the point of view of conservation of species. When people try to apply the Endangered Species Act, one of the most complicated types of problems that arise is what is the entity to conserve? And hybridization is the major reason why this question is often complicated. It's especially affects wolves, but also other species. There's an owl species, for instance, that hybridizes with another species that's not threatened. There are all sorts of interesting practical complications. So I think Javier's research will shed light to these questions as well and help develop our understanding. So with that, I'll let Javier tell the story.
All right. Thank you. Well, thank you everyone for coming. Thank you for the introduction. I know many of you came from a lecture that just finished a few minutes ago. You probably all lectured out, and I promise to make this as interesting as possible. I want to talk to you about my original research on admixture between coyotes and wolves, and possibly dogs are there also in the mix. Then I want to go into a brief tangent and talk about the genomics revolution that is going on right now that has facilitated my own work. And then I want to end with some more of a philosophical treatment of hybridization and its relationship to conservation, sort of what Rashid was alluding to just a minute ago. Well, I study northeastern coyotes, and when I speak to some people about what I study, I often get a response of surprise that, are there coyotes in New York? Really? Well, they are everywhere. They are everywhere in the East Coast except Long Island and New York City. Although a few have been spotted in Manhattan, there are no resident populations there. But basically how they got to the Northeast is very interesting. There were two waves of colonization. One north of the Great Lakes through Ontario. And because there are some resident wolf populations there, coyotes started mingling with them, hybridized with them, and so their descendants eventually made it into New York and colonized the rest of the Northeast. Roughly around the same time, there was another colonization as coyotes were moving eastward through Illinois and Ohio and Pennsylvania. But because there are no wolves south of the Great Lakes, those individuals that made it into western Pennsylvania and western New York were thought to be pure or free of admixture. So the genetic consequences of these two movements are illustrated in this figure. This figure is from a recent study of mitochondrial DNA. Mitochondrial DNA is a chromosome that is only inherited from the mothers. So what you see here is that in the Northeast, there's little genetic diversity, but about a third of individuals are carrying this chromosome from Great Lakes wolves, right? So this is evidence that there's a lot of admixture over here. In contrast, these individuals from Ohio have a genetic profile that is very similar to other typical Western populations, right? So there are no wolf sequences in Ohio. And in what the authors call the contact zones, where these two fronts of colonization are currently meeting in Western New York and Western Pennsylvania. So that's what we know about the genetics of coyotes, of eastern coyotes. We also know a little bit about their behavior and their morphology. See, wolves like deer, as in they like to eat deer, right? Coyotes in the West don't. They like to eat small things like roadrunners, rabbits, small things. But eastern coyotes like deer also, so they have a more wolf-like diet. Well, I want you to imagine this kind of extreme, bizarre situation. Imagine yourself trying to kill a deer with your mouth, right? Right? Imagine the serious muscles that you're going to need to keep this animal from escaping, right? Well, these serious muscles need some serious areas of attachment in the skull, and that's what you see here. This skull was prepared from a specimen in 1949 in northern New York, one of the first coyotes to be observed in New York coming from Ontario. This specimen is a more contemporary specimen from the same area. So I know it's only a sample size of 2, but in general, we know that over the last 7 decades or so, eastern coyotes have come to be not just bigger, but also badder. They have become more wolf-like. And I believe that this is an example of rapid adaptive evolution, possibly through hybridization, because through hybridization, all of these wolf genes, all of these wolf alleles or adaptive variants came into the gene pool of coyotes. And so these points of attachment in the skulls that we see in the contemporary eastern coyotes are associated with strong bite forces. And they're associated with resisting the mechanical stresses that are imposed by large, struggling prey. All right? Again, these skulls are more wolf-like. So I decided to investigate this issue of admixture, but remember that what we knew up to that point only came from one genetic marker, and it happened to be mitochondrial DNA, which is only inherited maternally. So I decided to investigate it from a different angle using many nuclear markers that are ancestry informative. And I did that in order to assess the prevalence and the spatial distribution of admixture, and also to estimate the coyote versus wolf ancestry in each individual, and also possibly to detect sex-biased hybridization. So I needed to select some genetic markers to do this, and when I was designing my study, when I was first starting out my research, this was around 2008, it was just a few years after the completion of the dog genome project. So maybe many of you have heard of the human genome project, right? Well, the dog genome project was finished in 2005. And with the completion of that project came the discovery of millions of single nucleotide polymorphisms, or SNPs. And SNPs are simply parts in the genome that if you were to compare 2 chromosomes, right? This could be 2 homologous chromosomes in the same individual or 2 homologous chromosomes in different individuals. But if in this position there's a C for cytosine and in this position there's a T for thymine, then that is a single nucleotide polymorphism. So these SNPs are quickly becoming the marker of choice among molecular biologists, especially among those who study species that are closely related to a model organism with a fully sequenced genome. So some of my collaborators from UCLA did just that, and they genotyped a large number, more than 60,000 SNPs in a wide sample of canids, including some dogs, western wolves, eastern wolves, and coyotes. And this data was recently published But before it was published, I had access to it, and what I did, I extracted from these 60,000 SNPs, 96, in order to do my dissertation, to explore these three questions, three broad questions that are part of my dissertation. But just for today, I'm going to focus on the 63 ancestry informative SNPs that I used to address this issue of admixture. So what do I mean by ancestry informative? Well, in any admixed population, there are 2 or more parental populations. And in my case, with northeastern coyotes, the parental populations are obviously western coyotes, eastern wolves, because hybridization, we know that hybridization occurred in Ontario with these Great Lakes wolves, but also possibly western wolves are also contributing to the ancestry of eastern coyotes. So I took this data set of 60,000 SNPs and I did a principal component analysis, or PCA. And a PCA is basically a way to visualize, in this case in 2 dimensions, a highly dimensional data set. Okay? So in this case, it was a 60,000 plus dimension data set. And this is just a way to reduce it into just these 2 axes of major variation. And what you see here is that individuals cluster by group, as we would expect. So western coyotes are genetically more related to each other than they are to eastern wolves, and so on. Now, but what's going on in the background of this analysis is that each of the 60,000 SNPs is contributing a little bit to the variation in this axis and a little bit to the variation in this axis. Some SNPs contribute more, some SNPs contribute less, so I ranked them by their contributions from highest to lowest. Okay? Separately, I did an FST analysis. And an FST is an index of genetic differentiation between 2 groups. So it goes from 0 to 1, 1 meaning that 2 groups are totally reproductively isolated. So basically, I did an FST between these 2 groups, between these 2 groups, and between these 2 groups. And I ranked the SNPs from highest to lowest, right? So now I have these 2 lists of SNPs, right? And I chose only those that were found in both lists. So now I have 2 separate but complementary tests from which I chose my genetic markers, and because they were strongly contributing to this variation, to this pulling apart of the clusters. Okay. So here they go. So here I chose 21 SNPs that are informative between coyotes and western wolves, 21 informative between coyotes and eastern wolves, and 21 informative between western and eastern wolves. If, let's say, I were to genotype one individual at this locus, right, and that individual had an A allele, we know, it got it from the wolf, right, because this is the wolf variant. Okay? So if you were to do that at a large number of SNPs like this, you could, in essence, estimate how much coyote is this individual, how much western wolf is this individual, how much eastern wolf is this individual. Okay? And I did this in a large sample of northeastern coyotes, 480 in total in the region. This is one of the largest molecular studies of coyotes to date. The one that I showed you previously was a study of 715 coyotes, but remember, it was only 1 molecular marker. So this was a study of 480 coyotes, but at 96 markers. So 17 of the 480 individuals totally failed. I got nothing out of them. That's not that bad because I had many left, 463, that gave me good data. So this is the number of genotypes that I expected, and I got more than 98%. So it was really good. The method worked very well, and I was very, very encouraged by that because there was no turning back, right? I need to graduate. So, so then I did a multidimensional scaling analysis of my data, and what a multidimensional scaling analysis is essentially very similar to a PCA, right? And so I have a data set with 63 dimensions, and I reduce it now to 2. And so here are all of my individuals in relation to the parental populations from the study of my collaborators. So here are the western coyotes, western wolves, eastern wolves, and then here are all my individuals. Now, two important things to note. This is an analysis of my SNPs, 63, and they provided a solution very similar to the genome-wide analysis of 60,000 SNPs. All right? That means that because these SNPs were so carefully chosen through these two tests that I explained, they did almost as good a job as the 60,000 SNPs, which was very encouraging. But biologically, what's interesting about this is that all of these guys seem to be admixed, right? They're intermediate between western coyotes and wolves. This is what we call a hybrid swarm, right? Where almost all individuals have an admixed ancestry. Very few of my individuals are clustering with western coyotes, and what was most surprising about this is that even in Ohio, where we thought the coyotes were pure and free of admixture, they were not. So the coyote individuals are here in the black dots. There's even one here that is kind of clustering closer to the wolves. So it's a very wolf-like coyote in Ohio, right? That was very surprising. Then I performed an ancestry analysis of my individuals in relation to the parental populations. So again, here are 36 western coyotes, 30 western wolves, and 13 eastern wolves. These are the parental populations. And then these are all my 463 individuals. And each individual is represented by a thin horizontal bar. And each bar is broken up into 3 segments, representing how much of its genome is attributed to one of the reference or the parental populations. So again, what was very surprising about this is that in Ohio, these individuals are far from pure, far from it. They're almost as wolf-like as individuals in the Northeast. So here in this table, it kind of summarizes the data. So in this analysis, the western coyotes came out to be 99.9% coyotes, of course. Western wolves are pretty much essentially just all wolf. Eastern wolves, all eastern wolf, right? But now when I assess admixture in my northeastern coyotes, they're all over the place. So Ohio coyotes are only about 64% coyote. That was very surprising. I expected them to have a genetic profile much like their western ancestors. So Ohio coyotes are certainly the most coyote-like, right? So they're 64% coyote, and northeastern coyotes are they're the most wolf-like, right? As we would expect. But this provides evidence for male wolves hybridizing with female coyotes. Because remember, in the previous study that I showed you, there were no wolf mitochondrial DNA sequences in Ohio, okay? But from this, from my study of nuclear genetic markers, there's a lot of wolf in these individuals, right? So so that wolf DNA had to come from male wolves, right? So this is evidence of male wolves interbreeding with female coyotes. All right, so this table is the one that I just showed you, just compressed this way. And the reason why I'm showing it again is because I thought that perhaps some of this signal of wolf may be dog. Okay, a lot of people kind of anecdotally say coyotes interbreed with dogs. They call them coy dogs in some places, right? And technically dogs are the same species as wolf. They're a subspecies. And so maybe some of this is actually coming from dog. And so I did that analysis. So I put 10 dogs into the analysis, and even though I did not ascertain the SNPs initially to distinguish dogs from other groups, the analysis did a good job at making dogs look like dogs, right? And so indeed, some of this got broken up into this, and a lot of that is dog, okay? So that was also very surprising because these proportions are non-trivial. Basically, almost 1/5 of the genome of these individuals is showing to be coming from dog ancestors. That was very surprising. So again, this is evidence of sex-biased coyote-dog hybridization between male dogs and female coyotes. In that previous study of the mitochondrial DNA, only 1 out of 715 sequences was dog. That's less than 1/7 of 1%. So we thought, oh, it's not important, right? Dogs are not important in this mix. But this is evidence that they are. So this DNA must be coming from male dogs. Okay, now when mammals hybridize, when different groups, different species hybridize, it's important who the mother is, especially in these large canines, because there is a lot of maternal care of the puppies. And so in this case, this is a wolf mother caring for her puppy. And I thought, could it be that individuals that have a wolf mitochondrial sequence, they are more wolf-like because they're going to be raised by wolf mothers, and they're more likely to back cross into the wolf population than into the coyote population. Okay? And so I did this analysis. So here on the y-axis is the coyote ancestry estimate from the analysis that I showed you a few slides ago. And here are I grouped individuals by whether they have an eastern wolf mitochondrial sequence, a western wolf mitochondrial sequence, or a coyote mitochondrial sequence. And basically, I only had these 2 individuals that were very wolf-like, and they indeed had a wolf mitochondrial sequence. So individuals with a wolf mitochondria tended to be more wolf-like. All right, that makes sense. But interestingly, individuals with an eastern wolf mitochondrial sequence were not very wolf-like. They were just as coyote-like as individuals with a coyote sequence. So I'm not sure why. Okay, then I thought, well, maybe since wolves like to eat deer, and so maybe individuals, these admixed individuals that are more wolf-like, they're going to like to hang out in places where there are more deer, right? So I had this deer map here, and these are all my samples superimposed over this deer density map. And actually what we see is that so here's the same axis, right, of coyote ancestry estimate, and these individuals that are inhabiting the most deer-dense areas, they are indeed more wolf-like, right? So they're less coyote-like, which means they're more wolf-like, right? So I thought that was interesting. So just to conclude this part of my talk, what we see here is a huge hybrid swarm. Basically, all individuals are admixed. Even in Ohio, that was the most surprising result. Even in Ohio, where we thought that these individuals were pure, they are far from pure. They are highly admixed. They're almost as admixed as northeastern coyotes. I also showed you that there's evidence for male wolf and female coyote matings, evidence for male dog and female coyote matings, and so there seems to be some sex-biased hybridization going on. And the fact that there was so much wolf DNA in Ohio makes me ask the question, well, how did that DNA get there, right? Did individuals circle all the way around the Great Lakes and come back to Ohio? We think that's improbable. We think that there may have been a separate hybridization event somewhere, probably in Michigan, but it's different than what we originally thought, that where it only happened in Ontario. It probably happened somewhere in the western Great Lakes region, and then the descendants of those individuals came down the southern Great Lakes into Ohio. Let me just go into very quickly what I intended to do next. Remember, I only showed you 63 of my 96 single nucleotide polymorphisms. I have 28 more with which I want to answer this question. Basically, how do different ecological factors influence population genetic structure in northeastern coyotes? And I have these other ideas about local adaptation. Basically, if I have some traits where we know the genetic architecture behind that trait, and I want to see if there are variations between northeastern coyotes and their western ancestors. So basically, I already genotyped these 5 genes, right? Some of them that are associated with skull morphology and body size, and these are some that I still want to do maybe after I graduate. But basically, I'll tell you really quickly about this one. Myostatin is a gene that is involved in musculature and basically in racing dogs. A group in Cornell found that one single nucleotide polymorphism makes a difference between a fast-running dog and a slower-running dog. And so I thought that this may be important in the wild, having this fast-running variant. We'll see. This is where I want us to go into this brief tangent about the genomics revolution that is going on right now. So the Human Genome Project was finished, or almost finished, in 2001. And after that, it wasn't the first genome to be fully sequenced, but it was the one with the highest profile, right? Got a lot of coverage. Most people have heard of it. And after that, all these other genomes started coming out, and I think this is in chronological order here. And so, okay. And so, with the publication or the release of these genomes, it just opens the door to ask so many more questions. Like I said previously, especially among those biologists who study the wild relatives of these model organisms. So when the dog genome came out, it opened a lot of doors to those who study wild canids, like coyotes, wolves, foxes, jackals, etc. The cat genome is almost finished, and so when that is finished, it's going to open up a lot of doors to those who study other wild cats, big cats, etc. And so a lot of that has to do with the profound and very sudden drop in costs of these types of projects. So when the human genome was finished in 2001, that project cost about $100 million, right? To do something of the same magnitude these days, it would take about $10,000. So if one of you has $10,000, right, faculty, right, you could get your own personal genome, all right? 10 years ago, $10,000 could only get you 1 million bases of sequence. 1 million bases of sequence now is free. No, I'm kidding. It's in the order of cents. It's like less than $1, right? So why am I saying this? Because if you're an undergraduate student, or if you're a graduate student early in your career, think about this. Think about how cheap you can get some massive data sets, how cheaply you can get some massive data sets and do PhD projects that are unprecedented. No one has done projects of this magnitude before. And so I started my studies somewhere over here. And what happened here was a radical shift in genome sequencing technologies from Sanger-based sequencing to what we call now next generation sequencing. But I started my project somewhere here, although I'm not doing sequencing technically. Anyway, I just wanted to spend the last few minutes talking about hybrids. All right, like Rashid was saying earlier, hybridization is a complex business. Fred Allendorf, who studies hybridized fish populations, said it is a complex and controversial issue, beginning with the task of identifying hybrids, right? Some notable biologists have even held hybridization culpable for the extinction of many species of plants and animals. So traditionally, hybridization has been put kind of on a bad light. But more recently, we've begun to appreciate the powerful evolutionary role that natural hybridization has, right? But then how do we distinguish between this natural hybridization and anthropogenic hybridization? So Allendorf and colleagues about 10 years ago attempted to categorize hybridization, right? Because there's obviously different types. But their first dichotomous separation is between natural and anthropogenic. Now, I think this is a useful exercise, and I think this is a good way to conceptualize hybridization, but implicit in the review and in the literature, there is this idea that natural is good. Right? I was wondering why he's so happy with a throbbing thumb. But natural is good, anthropogenic is bad. Okay? Anthropogenic is bad. You know how in Google, when you start searching like a term, it kind of completes something for you, like with some popular key terms. So yesterday I was typing anthropo, and before I even finished, it's like anthropogenic climate change, anthropogenic pollution, anthropogenic disturbance, anthropogenic everything bad, right? Like, is there anything good that we do as humans? Like anthropogenic chocolate brownies, right? I mean, my baby is anthropogenic, right? So there's this philosophy that this is bad, and this makes its way into policy, right? Landmark legislation in the United States, the Endangered Species Act, the ESA, 1973. It was a great piece of legislation designed at protecting species. But hybrids are not mentioned at all. There's just, it wasn't even, it's not there. So in the 1980s, when all this evidence of admixture started coming up, like, oh, what do we do with hybrids? And so the policy before 1990 was they don't warrant any protection, okay? Then in 1990, the US Fish and Wildlife Service said, well, let's rethink this. These standards are too rigid, and we should revisit the situation. So I guess they revisited, or they thought about it for 6 years, because it wasn't until 1996 that the intercross policy was proposed, which allowed the flexibility to deal with things kind of case by case, right? Not saying let's not protect hybrids, not saying let's not protect hybrids. It was a very flexible policy, but to date, it has neither been approved nor disapproved. So I know the government takes a long time in making decisions, but 15 years, and there's still no policy guiding what to do with hybrids. Now, maybe we don't need a policy. That's okay too. But I wanted to bring this up. The U.S. Endangered Species Act is about protecting species. But what are species, right? So if you're a biologist, it's not surprising that there is no uniform concept of what is a species, right? There are many definitions of species. Okay, so if we go into the actual document, the Endangered Species Act, maybe it'll say there, but it doesn't. So the Endangered Species Act doesn't define a species either, so there is also no legal definition of species. All right. Now, we know that the Endangered Species Act is kind of biased toward the vertebrates, although it also protects invertebrates and plants. And we also know that the Endangered Species Act is kind of biased toward adopting the biological species concept, which says that two populations are distinct species if they are reproductively isolated. But in my case, with canids, here's the western wolf, the eastern timber wolf, the Mexican wolf, the red wolf, here's a domestic dog, a coyote, very urbanized coyote found in the train, and our very own, the sea wolf. No, I'm kidding, the sea wolf is fictional. But we have all these canids in North America that they're not reproductively isolated at all. In fact, they interbreed very freely, and their offspring are very fertile, and one can argue that their offspring are actually have a higher fitness than their parental population. So basically, by the concept that we like to think, many of us like to think about, these are all just one species. But right now they're taken as different subspecies and different species. But I kind of argue that they're all one species. Okay, anyway, let's have a conservation conversation. This is where I hope to get a little bit of audience participation, just for like a minute or so. I mean, what do you think about what I'm saying? Should we protect natural and not anthropogenic hybrids? Please, I want to hear your comments. You know, often when these things are are included in policies, the government opens them up to public comment. So let's imagine that we're in that situation. What do you think? Yes.
Are there other families where this happens as well? Is it common in the cat families to have this sort of same genetic mixture?
That's a good point. So the domestic cat, there's debate whether that is its own species or is a subspecies of the wild cat. And so in Europe, they also seem to be dealing with the same situation where domestic cats are hybridizing with wild cats. And so you have a hybrid swarm, essentially. Yeah. So should we not protect then these admixed cats? Yeah, Matt, what do you think?
So I'm not going to stand but I will say, how are you going to define natural versus anthropogenic hybrids? I mean, is the coyote a natural hybrid, or is it anthropogenic because we hunted out all the wolves that were keeping the coyotes from invading the east?
Okay, that's a great point, because that's what I have here. What if we are several steps removed from the issue? Right? It's not like we translocated the individuals themselves. I mean, that's happened. Like when you put like a trout from an eastern river and put it in some river in Oregon, and then they're going to hybridize. That's like by direct translocation. But we think that coyotes just made their way eastward because we denuded the forests in the Great Lakes region, right? And coyotes like open areas, and so now what was formerly forested was now open, and then they just made their way, met up with some wolves there and hybridized. Yes, Diane?
But domestic dogs are a totally different issue. And virtually all of your animals are 20% domestic.
Yeah.
So how does that fit into this?
Right, this is why it's a complex business. It's, yeah, I don't know. This is all new, right? That these individuals have such a high proportion of dog in their genome. But then are dogs not natural? You know, and so we get into this kind of philosophies, right?
But domestic, but it goes to the whole question of domestication, right?
Yeah.
So once you domesticate species, are domesticated species anthropogenic?
Right, Doug seems to think so, yeah. So yeah, they are, but I don't think they're bad, right? Because they're anthropogenic. All right. So right now there's a very aggressive policy by the U.S. Fish and Wildlife Service to remove hybrid individuals in the red wolf recovery zone. Okay, and so this brings up the question: is it ethical to just poison or shoot a hybrid coyote red wolf? All right, so this gets very hairy if we start going really into it. But thank you for your participation. I just want to end with this. Basically, what are we trying to conserve? Well, in my honest opinion, we should be trying to conserve natural evolutionary processes. Here we go again with natural. But we need to understand that evolution doesn't happen in a nice, neat way that our models kind of make us believe it happens, right? Where one lineage splits into two, and this lineage splits into two, and this one also splits into two. But sometimes these lineages that split into two, they merge back together. And so evolution can also be reticulated, not strictly bifurcating. And we also need to remember that biodiversity is not something that's static. It's always something that's moving and constantly evolving. And I also want to point out that we need to strive to conserve healthy ecosystem functions, so that in some respects it doesn't really matter if individuals are hybrids if they function in the same way as their pure parental populations did at some point. So for example, New York is now repopulated with these Coyote-Wolf hybrids, right? It doesn't matter that they're not the original eastern timber wolf that used to live here before the 1800s. Well, there's a huge deer overpopulation problem. There are more than 1.5 million deer vehicle accidents each year in the United States. More than 50 of them in New York alone, right? Many people are seriously injured or die in these accidents. And when cars become the primary control for deer populations, we have a problem, okay? It's a huge societal cost. Let me say that again. When deer populations are controlled by cars, the costs are paid by all of us through our insurance premiums, right? We need to restore healthy ecosystems by having top predators, and and I think that the coyote-wolf hybrids that are currently inhabiting the Northeast serve that function. Well, I want to end here by thanking my advisory committee, Dan Dijkhuizen, Catherine Graham, Rashid Achikaya, and my outside committee member, Roland Case from the New York State Museum. Here are my funding sources: Turner Fellowship, the NSF, American Museum of Natural History, American Society of Mammalogists, New York State Museum, and the NIH. And I didn't do this by myself. I had a lot of help. And I lastly want to thank you for your attention. Thank you very much for also your participation. I'll be glad to take any questions if you have any. All right. Yes, ma'am.
So, the different pictures you get from looking at mitochondrial data and food data is very interesting. And from that, you can draw the conclusion that the hybridization events were male wolves and female tigers.
Correct.
But so all the kind of scenarios we talked about have been very behaviorally moving. Think of a situation where rather than behavior, the direction I'm taking, if you have a female wolf and a male tiger, and then you have infertile hybrids, infertile female hybrids, or reduced viability of the female hybrids from that cross, you still get a same picture of the kind of different picture of the mitochondria.
Okay.
And I don't know much about physiology of wolves, but I imagine there's quite a bit known in domesticated dogs, and you can maybe draw some conclusions from looking at that literature. It would be interesting to see.
Yeah, no, that's a good point. When two groups hybridize, the viability of the offspring sometimes depends on who the father was or who the mother was, right?
Yeah, and
so
the sex
that's,
of the offspring as well.
and the sex of the offspring as well. That's right. I don't know if that applies here. I'm not sure, but definitely the coyote-wolf hybrids seem to be doing very well, and I argue that that is because of the introduction of adaptive variation. Let me just say one thing, that the one instance of dog mitochondria found in a sample of 715 coyotes could be sampling bias, right? Because if female dogs are mating with male coyotes, those puppies are going to be born to dog mothers, okay? And they may not make it into the sample, right? Because they're going to be born in someone's backyard or something like that. Okay. Diana, did you have a question? No? Okay. Oh, Matt.
So you did regression, more or less, regression of amount coyote versus wolf on the deer. And I noticed in your map that far northern areas have lower deer density than, say, New York or Pennsylvania.
Mm-hmm.
But my understanding is that there are other prey in those areas. I know in New Hampshire, at least, coyotes have been observed to take down young moose as a pack, and not as an individual, which is
Sure.
a behavioral difference, obviously, between moose and coyotes. So do you think your patterns would be any different if you accounted for other large prey other than just deer?
I think so, but I don't have a way to do it quantitatively. I don't have a map of moose deer density, and even if I did, it would just be restricted to the far portions of the extent of my study, right? So it would be like, like presence or absence, and then
more cumulative, like large prey rather than deer per mile, like large prey per mile.
yeah, that's a good point. So the deer, the availability of the deer density data is only because deer are very aggressively managed in most states, and so each state has like a deer biologist, right? So, yeah, but that's a good point. Yeah, Dan.
Actually, I have a question. So the morphological difference between your two skulls is really interesting, but we also know from lots of other studies on other vertebrates that they have what's called, use-induced hypertrophism, right? So the harder the food you eat, the more it pulls on the muscles. The more you pull on the muscles, the more it remodels the bone. So do you have any idea how much of that morphological difference is actually plastic due to the fact that they're eating more deer, chewing on harder things, and so their face gets fatter?
I don't. That's a really good point. It's possible that the morphological differences are due to phenotypic plasticity. They're not mutually exclusive, right? It could be both genetics and plasticity at the same time. But that's a good point. I don't know if it could be that. Yeah, so I mean, some people have argued that coyotes in the east are larger because of there's more food. Yeah. I think these molecular studies really start to address the issue. Yeah. Yeah, Adam?
Do the coyotes in the west harvest frequently with dogs, or have there been studies done in the native regions?
Okay, very good question. So is there hybridization in other regions? Because coyote range is now very expansive, right? No, I don't think so. And so this is one of these interesting things that only occurs in the east. Coyotes and wolves in the west are very antagonistic against each other, especially wolves against coyotes, right? Because their size difference is greater. And so we don't know why this is only occurring here in the northeast. And it doesn't seem that dogs are contributing much. That paper by on the 60,000 SNPs, it showed that dogs are mainly contributing to the genomes of eastern coyotes, not to western coyotes and not to western wolves. Yeah.
Let's
All
thank
right.
our speaker once again.
All right, thank you very much.